Welcome, everybody. Thank you so much for joining us today. I'm just letting people in from the waiting room as we kind of kick off today's meeting. If you don't know me, if you're not part of CTF Engage or we haven't had the pleasure of meeting, my name is Kate and I work with the Children's Tumor Foundation in patient education and engagement. One of the things that I do is run our patient engagement program, which is called CTF Engage. And it is in some ways very similar to RAINS, in other ways a little bit different. And it's been a lot of fun to work with Vanessa Merker and to kind of learn from her and everything that the RAINS group has been doing as we've gotten this started. So I'm happy to have both our RAINS folks and our CTF Engage folks here with us today. So I'm going to do some housekeeping and then I'm going to get us right into our fabulous content for today. So first of all, just make sure that you are muted unless you're asking a question at the end, just so that we don't have any interruptions for our speakers today. Closed captions are available by clicking the CC button at the bottom of your screen. We have a live captioner here today, and if that's not working for any reason, be sure to talk to me in the chat so I can make sure we get that corrected. And make sure that, also, I would love it if you would introduce yourselves in the chat, since we do have a mix of folks today, not just our CTF Engage group as we generally have for our meetings. So if you'll just drop in maybe where you're located and your connection to NF or CTF or RAINS, that would be really a great way to kind of get to know each other a little bit. We are also going to be recording today, so if you don't want your image recorded, you're welcome to turn off your camera. And we will save time at the end for a Q&A with our speakers. So you're welcome to put questions in the chat as you're thinking of them and listening to the presentation, or if you're just more comfortable asking them there and don't want to ask, on the live call, please feel free to use the chat. Otherwise, at the end, you'll be invited to unmute, raise your hand, unmute, ask a question, and we'll take some time for that at the end of our presentation. So having said all of that, I will say that I am very excited to welcome our speakers for today. So we have Miranda McManus and Kara Anstedt, who some of you may know already. They came to the NF Summit in 2023 and spoke on this topic and did a really fabulous job. And they have more to say, and we're really happy to hear from them. So Miranda is actually a parent to a son with NF1, her inspiration and hero. She's also part of the biology faculty at the College of Charleston in South Carolina and has a background in molecular genetics. She's a member of the RAINS Gene Therapy Working Group and serves on the RAINS Directors Council. Kara is a board-certified genetic counselor with a specialty in neurology and has worked in the NF and schwannomatosis field since 2015 at the NYU Langone Health Comprehensive NF Center, which is in New York. She's a clinical supervisor and a guest lecturer for multiple genetic counseling programs, and she participates in multiple international collaborative efforts in neurofibromatosis, including the NF Variant Curation Expert Panel and the RAINS Gene Therapy Working Group. So having said all of that, I'm pleased to hand things over. We're going to start with Miranda, who is going to kick things off for us. Thank you, Miranda.
Yeah, hey everyone. Thanks for having us. Get this on the right screen. So there's a lot of talk in the community about gene therapy, right? You're starting to see drugs approved for other conditions that involve gene therapy or protocols that involve gene therapy. And it's really important. We aren't there in NF yet, but it's really important to make sure that we as a community are getting prepared for this, because there is a little bit of basic knowledge to understanding more about gene therapy. And so that's kind of what Kara and I want to get into today. I'm going to start off with just some of the basic biology, talk about what genes, DNA, chromosomes, all these things actually are, and then how the genes that are involved in NF and schwannomatosis are impacted in somebody with schwannomatosis or neurofibromatosis. And then Kara is going to move over into talking a little bit more about where we are in NF and the different types of gene therapy, because this is really broad. So you'll notice on our title slide, we said gene targeted therapy. As our RAINS group, we've been really focused on trying to be all inclusive about this topic, because it's not always the genes that are being altered in any way. And so we need to kind of understand the basics in the way genes work within our bodies. So our genome, which includes like all of our DNA makeup, that's like a general instruction manual, right? It tells us all of the things that need to be made in our body, how they work. But there's a whole process before those genes actually get functional, right? Every single cell in our body has the same set of DNA in it. And that DNA is turned on and off in those different cells to do different things. So the way that our DNA is arranged in humans is that we have 23 different pair of chromosomes. So 22 of those are like the exact same chromosome that may have different information on them, but they code for the same things. And then the last pair are sex chromosomes. And so, those are going to appear different in males and females. And so they don't have necessarily the exact same genes on them. But for NF, that's not very relevant because the gene for NF and schwannomatosis are both found on the other type of chromosomes, not the sex chromosomes. So those chromosomes are basically just super long DNA molecules that are condensed down and stored in the nucleus of our cells in our body. And these long DNA molecules are made up of different smaller molecules that go together and function kind of like an alphabet. So the letters that you'll see in DNA sequences are A, T, C, and G. It actually stands for the things that are sticking off in the middle there. And then groups of those letters end up being our genes. So a gene might be more like a word, or a set of genes you might think of as like a sentence, right? So all of this is made up of DNA molecules. Chromosomes have a lot of different genes on them. When we're talking about a gene, we're just talking about one segment of a chromosome, all right? So just to think about this in the broader context, there's 3 billion of these letters about in our genetic, in our human genome. And there's about 20,000 different genes, and some of those genes can actually be used for multiple different things. And that's all on these 23 pair chromosomes. So there's a lot of information there and a lot to really sort out and understand. So if we're looking in this example at NF1, but we see the same type of inheritance pattern in NF2 related schwannomatosis and in other forms of schwannomatosis, it's what we call an autosomal dominant condition. So what that means is that it's on one of those chromosomes that's not a sex chromosome. That's where the autosomal comes from. And it's dominant because each of us has pairs of chromosomes that's essentially one from our mom and one from our dad, right? So each of the chromosomes are going to have all of the same genes on them, but it might have different versions of those genes that code for different things on different chromosomes. So with NF or schwannomatosis in any form, the mutation or genetic variant is the terminology that's used a little bit more commonly now in humans, that causes the condition is only going to be on one of these chromosomes. All right. So it's either the one that came from mom or the one that came from dad. It doesn't mean that you inherited it from mom or dad, because this happens sometimes just spontaneously, about half of the time, in the case of NF1, for example. So in this case, we've got dad over here that has one affected chromosome and one unaffected chromosome at that gene that causes NF1. And we've got mom that has 2 unaffected chromosomes. They have the wild type or normal version of that gene, not the one that alters the gene and causes a problem. So each of these individuals, when they have kids, is going to pass down one of those chromosomes. So you see those are noted by being blue or pink. And so half the time, dad over here is going to pass down the affected chromosome. And so out of these 4 children, 2 of them would be affected in this case by NF1. All right. And it's all because of some alteration in this gene. So we're going to zoom in a little bit more. all right? Because to understand gene therapy, you really need to understand that genes have the instructions, but that's not what ultimately does the work within our bodies. So genes are actually made up of subunits as well. So you can see in this case, we've got a bunch of exons and introns within this gene. Remember, the gene is just a portion of that DNA molecule, all right? So the exons are the functional part of the code here. The introns actually end up getting snipped out later on. And it does it by going through this set of processes that we call transcription and translation, where the DNA gene gets transcribed into a very similar language of RNA. It's a molecule that's very similar to DNA, but it cuts off these ends. So there's some other parts. So it makes a shorter version of the gene that is just the gene, right? And then it goes through another stage where all of those introns get sliced out, and you end up getting this messenger RNA molecule, which probably nobody had heard of 4 years ago, but now everybody's at least heard of messenger RNA because of the messenger RNA COVID vaccines. So this messenger RNA molecule is essentially what gets that code from the gene out of the nucleus, because DNA never leaves the nucleus. It stays tucked away safe in the cell. So this messenger RNA code goes out just with the exons in it. And then ultimately goes through this process called translation that ends up creating this amino acid chain, which is what makes up proteins. And so there's different proteins that are affected in NF and NF1. We've got neurofibromin, right? So these different these proteins are ultimately what functions within the human body. And so if anywhere along this process something is altered, it can create a protein that's potentially not functional, right? Or not functioning well. So the NF related proteins do things within our bodies like preventing cells from multiplying too much. It functions as a tumor suppressor protein, which basically shuts down cell division before it goes too far. They're also used in nerve cell signaling and in controlling other genes that are related to pain and inflammation. So sometimes these proteins have multiple roles. And in the case of neurofibromatosis and schwannomatosis, there tends to be a lot of roles that impact multiple body systems. So I wanted to talk about a little bit about the different types of genetic variants that we have. So the way that DNA is read is actually in sequences of 3 letters. So remember those A's, T's, C's, and G's? They're arranged in sequences of 3 letters that ultimately end up coding for that amino acid and then the protein. And so it gets read in groups of 3s. And that's pretty important when we think about the different types of genetic variants that you can have. So I think it's easier to see it in the form of a sentence. So we've got a bunch of 3-letter words here in our original or our wild type gene here. So this would be the red cat ran out the car. So we have various different types of mutations. One of them is a missense mutation. So in this case, the red cat ran out the bar. Still a sentence, might even sometimes make a little bit of sense still. Might not mess things up too bad, but can potentially cause a big problem as well. And in NF, we see missense mutations that can essentially completely shut down the function of the gene. So it depends on where that is as to how much it matters. We can also have insertions or deletions. There's 2 different types. So an in-frame insertion or an in-frame deletion is going to be in a group of 3. So in this first sentence with the insertion, it altered it to the big red cat ran out the car. Again, might still make a little bit of sense. It might cause a problem, but it might not be a huge problem if it's something that fits in with everything else. And the same thing with the deletion. The cat ran out the car, and it doesn't mess up the rest of the sequence because it's in that group of three, right? Whereas in a frame shift insertion or a frame shift deletion, it's a multiple of one or two rather than of three. So you can see if we just add a B here, it doesn't just impact that one word, it impacts the entire rest of the sequence. So sometimes, a single letter insertion can be devastating, right? Because it messes up the rest of the sequence. So you think if this happens early on, then it can definitely impact the function of the protein. And then the last would be a nonsense mutation. So a nonsense mutation is something that codes for a stop codon early, which means that something that should code for a word has been changed to where it now signals the termination of this process. And so it might get cut off right there. So in this case, right after the cat. So those are just looking at a few letters at a time. Genetic variants are not always confined to a small amount. So there's different things that can happen. And just because of natural processes and cell division, some mistakes can be made sometimes and lead to things like duplications or deletions. So you can think of this as being different genes within this chromosome. And in this case, for some reason, this E and F ended up duplicating. We end up getting a lot of duplications with our chromosomes over time, right? And then once that duplication is there, that can potentially get passed down to the next generation as well. So it creates different versions of genes. If it's within a gene, or more copies of a gene if it encompasses more than one gene. So we could see the same type of thing where deletion. So a deletion could just be a couple letters in the DNA sequence, or it could be a couple of genes or one gene. We can have inversions. Sometimes they get turned around. So then they're completely backwards within the genome. Or we can even get translocations that could potentially happen within two different chromosomes, where a set of genes might jump over to a completely different chromosome. And in this case, this is what we would call a reciprocal translocation, because you notice the EFG and the QRS just swapped. So sometimes that happens, sometimes it doesn't. So all this just to say, there's a lot of different types of genetic variants that can occur. And in NF and schwannomatosis, we're dealing with large genes. And so we can see a lot of these different types of mutations that have occurred within those genes. You probably see this in a slightly different form. If you get a genetic report, right, it might have your DNA sequence showing your genetic variant, and it might look something like one of these. This is just notation for describing different things. So in this case, this is a change of a single nucleotide to another. And in this case, though, that ends up changing that particular three-letter codon into a stop codon. And so that's a nonsense mutation. Remember when it gets cut off. We see these in NF and schwannomatosis. This is a truncating type of mutation. So there's no functional protein that is likely going to be made from something that's truncated, because it cuts off the rest, right? We can also see deletions that are marked by this del in here. In this case, it's a frame shift deletion. So I'm not going to get into all this notation because that's just going a little bit too deep, but just so you understand, that's what these are coding for, right? This is a splice site. So splice site mutations impact that exon intron border. And so sometimes something that is supposed to get snipped out doesn't get snipped out, or something that's not supposed to get snipped out gets snipped out. We can have missense mutations that just code for another amino acid in that protein sequence. So sometimes that can be a problem. Sometimes it's not as much of one. And then we can have deletions where like an entire exon is removed, right? So again, it's not just a single letter or gene. It can be in any number of different types of deletions. And then many of you have probably heard of the micro deletion in NF1. We can have whole gene deletions where you're not going to get any functional protein out of that, right? So there's a lot of variety in the different types of mutations that can happen. But in NF or in schwannomatosis, we're going to see the same type of thing. Regardless of the mutation, if you have a mutation on one of your chromosomes or a genetic variant on one of your chromosomes, then when that is transcribed into RNA, that variant is also going to get transcribed into the RNA. And if it's transcribed into the RNA, that RNA code is what's used to make the functional protein. And so you might not get the functional protein for half, right? We've got the other one. And that's the reason we only see NF or schwannomatosis when it's just a single chromosome that is impacted, right? If it's the other one too, it's going to be too much on your system as a germline mutation. And so that individual would have never been born, right? But if you have NF or schwannomatosis, then you have a deletion on, I'm sorry, not a deletion, a variant on one of the chromosomes that gets passed down into the RNA, and then will either make no functional protein for that one copy, or you might get one that doesn't work and so isn't functional. So there might be a protein, but it might not be functional, right? So these germline mutations, we call them, the one that you're born with, right? Not mutations that may happen later on in life, but this germline variant causes some of the non-tumor features in NF1. So this is implicated in things like developmental delays, learning disabilities, scoliosis, vascular disease, some things that generally go along with NF1, possibly. It can also cause non-tumor features in NF2-related schwannomatosis like cataracts, and can also play a role in chronic pain. All right, so oops. So I'm going to stop my share there and pass this off to Kara for her to pick up the other half.
So Miranda started by talking a lot about our germline variants and how they play a role in certain symptoms related to NF. But as many of us know, NF and schwannomatosis is primarily defined as a tumor predisposition syndrome. And so what happens to cause these tumors to develop? And so what happens is we start off with all the cells of the body have this single mutation in one copy of the gene, and the other gene works correctly. And what happens is in even just like a single cell, we get something called a somatic mutation. So a mutation that happens after you're born in some part of your body is called somatic. If that person has a diagnosis of NF1 or an NF2 related schwannomatosis, it's just one mutation in the other copy of the gene. If they have other non-NF2 related schwannomatoses, there's actually a couple different mutations that happen. It happens in the other copy of LZTR1 or SMARCB1. And then it also happens in a way that mutates the NF2 gene as well. So all types of schwannomatosis within the tumors, we see NF2 gene mutations. And so that happens in the other copy of the gene. It's a random event. We're always experiencing things in life that mutate our DNA. But sometimes it can be triggered by things like radiation from radiation therapy, we know can cause mutations in DNA, but most of it's just a random event that we don't have control over, these mutations in single cells. So it happens in that other copy of the gene. And thus it's in the RNA that's being made from that other chromosome, and then results in also a mutation of some kind impacting the protein on that other copy. And so you end up with a cell that doesn't in NF1 have any functional NF1 protein, In NF2 schwannomatosis, no functional NF2 protein. In the other types of schwannomatosis, the NF2 is not working, and the other type of protein as well. And so that cell, it's missing these things that are really essential for how it works, and that starts to cause different changes in the cell. And what it primarily causes is, because these genes are so important in controlling cell division, is it causes that cell to start to divide more than it should. And any cell that divides faster and more frequently than it should can become a tumor over time. And so that's where we see the different benign tumors of NF and schwannomatosis, like neurofibromas and schwannomas, meningiomas, optic pathway gliomas. Those are all related to these multiple hits within a cell, and then that cell starts to grow and divide. So that's what happens if it's in a nerve cell. If that random second or multiple mutations happen in the pigment cells of your skin when you have NF1, that's what causes the cafe au lait spots and the skin fold freckling, the increased pigmentation in the skin. Particularly for NF1, we also see a risk of cancerous tumors. And so particularly things like MPNST or a high-grade glioma, what we think causes that is you start off with the mutations within the NF1 gene, and then more mutations happen later within that same cell, those same cells, that same tumor. There's a couple genes we've identified that play a role in turning a benign neurofibroma to a cancerous MPNST, like SUS12 and CDKN2A. So more mutations are needed. But there are these somatic mutations that happen within a tumor that cause that, within cells that cause a tumor to develop. And understanding what causes the non-tumor features and what causes the tumor features of NF and schwannomatosis is important in understanding how we might try to target it with different gene-directed therapies. So I'm going to kind of walk through different places where we might be able to try gene-directed therapies and kind of different things that we know about them in the context of NF and schwannomatosis. So one of the most studied forms of gene-directed therapy is what we call gene replacement therapy. And so that's when they use a viral vector that contains with it an extra sequence of the gene, a working copy of NF1, for example, or NF2, and it delivers it into your genome and kind of inserts it into your DNA. So you get like an extra copy. So the mutation is still there, but you have this extra copy that's a working version of the gene. These are usually permanent types of gene therapy because it gets inserted into your DNA. So it's just a single treatment that causes it. It's very well studied how we can do gene replacement therapy, and it's used in some diseases, but it can be challenging. One thing that is tough about gene replacement therapy is there's a limit to how big the gene can be with our current technology. So it's something that might be harder to do for NF1, for example, because the NF1 gene is one of the biggest genes in our body and is in some cases too big for the types of viruses that are used for gene replacement therapy right now. Another DNA level therapy that you might have heard about in the news, a lot of people talk about CRISPR now, is gene editing. So what that does is it targets the mutation that's within your germline DNA and then corrects it. So it removes the mutation and should convert the gene back to the functional wild type copy without the mutation over time. And because this is on the DNA level, again, this would be a permanent change in someone's DNA and probably would just be a single treatment to do it. We can also do gene-targeted therapy on an RNA level. So we don't touch the DNA at all. We don't mutate the DNA. We don't add anything to the DNA, but instead, a treatment is given that modifies the RNA that's being made by the mutated gene. RNA is something that's constantly being made and then like recycled over and over. So you make RNA, you use it to make some protein, and then RNA gets broken down. And because it's something that's constantly being made and broken down, anything that targets the RNA as a treatment option is going to be something that's temporary, and usually something that we're going to have to give someone as a treatment on a regular basis. So you're going to have to keep getting the RNA-based therapy because it's just going to flush out of your system over time. Exon skipping is a type of RNA therapy to skip over and remove an exon that has a problem in it. RNA editing to fix the mutation within the RNA, leaving the DNA alone, is also something that's in development for different types of conditions. Sometimes in gene-targeted therapy, we might look at, we're not going to do anything in the DNA or the RNA, but maybe we figured out how to give you extra neurofibromatosis protein if you have NF1, because we know that's the thing that's doing the job in the body. And that is used in certain types of diseases. It's a little bit challenging in NF and schwannomatosis because of what we know about these proteins. So it could be something that's a little bit harder for us to develop right now because of how it functions in nerves and how we can isolate it and also get it where it needs to go. So probably not the best option for NF and schwannomatosis, but it is used in other diseases. There's also something called nonsense suppression. So if you have one of those nonsense types of mutations that cuts off the end of the gene and prevents the protein from being fully functional, there are sometimes ways that you can kind of put a Band-Aid on that nonsense variant and prevent it from malfunctioning in the body. So that is sometimes used if people have a very specific type of mutation. The things about these gene therapies as well is some of them are custom made, very unique to the mutation that that person has. Some of them could be used for many different types of mutations. So it could be used for many people with a single type of treatment. So gene editing is usually custom to that very, very specific mutation that someone has. That can be challenging because many people with NF and schwannomatosis have very unique mutations anywhere along the gene. So customizing a CRISPR method in NF and schwannomatosis might be harder to do because it would have to be made unique for each person. Gene replacement therapy, though, it doesn't matter what type of mutation you have because you're getting a brand new copy of the gene added in. So that one is kind of more generic and can be used for many different people regardless of the type of variant that they have. And then lastly, there's one that doesn't even actually target specifically your NF or your schwannomatosis mutation, but there is something called suicide gene therapy. And so what that does is actually very specific to tumors and targeting the genetic mutations within a tumor that cause that tumor to grow and develop. Sometimes it's actually inserting a completely different gene that goes and kind of mucks up that tumor in terms of how it's operating and can cause the tumor to shrink and start to the cells to start to like die off within the tumor itself. So that's sometimes used and is being investigated in different tumor and cancer syndromes is, can we introduce extra DNA that prevents a tumor from continuing to do what tumors do, grow and divide? So one other thing that we have to think about with gene therapy is how do we get this treatment to where it needs to be in the body? And there are different types of ways that gene therapy can be delivered in different research settings and in different diseases. There's lots of different methods that are being done because we need to balance getting the gene therapy to where it needs to be so it can be effective, so it can get into all the cells it needs to be so that it can start operating, while also not causing excess side effects and other issues with other parts of the body. And so there are what we call systemic therapies, so where the gene therapy is spread throughout your body. For example, by IV intravenous, so into the vein, you have the gene-directed therapy that gets into your blood system and kind of spreads through your body like that. And that could be advantageous because if it's spread throughout your body, then it could potentially act on a lot of different body parts at once. And so it could treat multiple symptoms at once. We can also have what we call local treatment delivery options. So rather than kind of spreading the gene therapy throughout your body, let's target a very specific body part or tumor or organ where we really want it to operate. And so there are certain gene therapies in other diseases in which the primary treatment is we want to treat something happening in the brain, a brain tumor or a cognitive issue. And so there are gene therapies that involve an injection into the brain in order to get it really, really high concentrations there. There are also some types of gene therapies that are injected through the spinal cord so that they can spread throughout the spine and into the brain as well, because that's all connected. One thing about the brain and the spinal cord is it's really good at keeping things out, used by something we call the blood-brain barrier. And so that's why sometimes treatments are injected directly into those parts of the body to kind of get around that protection that we naturally have. For NF and schwannomatosis, we might also be talking about something that gets injected directly into a tumor. So something that we want to act locally within a tumor, like in a plexiform neurofibroma, if you have NF1, or maybe specifically into certain schwannomas, that might also be a way that we can target directly into a tumor that we want to treat. As I mentioned, depending on what part of the DNA, RNA, protein pathway a specific gene therapy is acting, gene therapy might be just a single one-time treatment with permanent effects, or it might be something that has to be given continuously. So on a regular basis, you have to go in and have the treatment so that the gene therapy can act within your body because it gets broken down. And then when we think about treatment goals, gene therapy is in really early stages right now. And so probably the most immediate goals when this starts to get into human clinical trials would be, can this gene therapy slow down a symptom of some kind or a clinical feature? Can it slow down how a tumor is growing, for example? And then from there, as it develops, we'd be looking at things like stopping something from happening. So can we stop the level of severity for some symptom where it is right now and prevent it from getting worse? And then eventually we're hopeful that this can be used as a preventative kind of treatment. So can we prevent different clinical features and different tumors and different medical issues from NF and schwannomatosis developing in the first place? But traditionally with clinical trials, we start with, can we treat what someone already has and slow something down or stop it? And then later trials look at things like prevention. So this was the status of kind of gene therapy when we initially gave this talk in 2023. So the green symbols indicate a gene therapy that is available on a clinical FDA approved basis in some sort of disease. None of them are approved in NF. None of them are in human clinical trials for NF and schwannomatosis, but there are ones that have gone through all the clinical trials and are now approved in other diseases. Those include gene replacement therapy, exon skipping for certain diseases, nonsense suppression, and protein replacement. And I put the diseases that we have seen, some examples of conditions that have this approved. CRISPR is really in a research setting for a lot of diseases right now. So is RNA editing, as well as the suicide gene therapy option. And last year, some of these were being worked on in different models. So we had some mice studies that were ongoing for gene replacement therapy and for nonsense suppression. And then a lot of it is really looking at cell cultures. So not even in animals yet, but just those initial stages of, we have a petri dish of cells. Can we get the gene therapy to work within that before we move on to animals? So at the most recent NF meeting that was in Belgium, the global meeting in June, there was a lot of talk about gene therapy and where we're going with this. There's a lot of excitement over what we can do with it. And there were presentations from different researchers about results from studies that they were working on. So I have some really brief summary about the things that were talked about at the meeting. So there was a presentation on NF2 gene replacement therapy. The NF2 gene is a lot smaller than the NF1 gene. So it is one that could work really well for gene replacement therapy. And it was a study, a really kind of initial study looking at different versions of the viral vector and the gene sequence for NF2 in a bunch of different models. So they looked at mice, that had schwannoma tumors, they looked at some different cells from people with NF2, from vestibular schwannomas, and even from a cancer called glioblastoma. And they treated all of them with these candidate gene replacement therapies. And they did see that the one that they had designed was able to increase the amount of NF2 RNA and protein that was being made. Sometimes as much as the kind of like what we would expect in someone who didn't have any NF2 mutations. So kind of what we called rescued the protein and the RNA. And when it was given to the mice that had schwannoma tumors, it made their schwannomas shrink. So really promising initial studies for this type of gene therapy. And as with gene replacement therapy, that's usually something that's permanent. And interestingly, the NF2 gene has a lot of function in other types of medical conditions. And actually, NF2 mutations are seen in a lot of different cancers that are unrelated to NF2. And they did initially check it in some cancer cells, and it seemed to have an effect. So it's kind of an opportunity for gene therapy that could be helpful for people with NF2, but also helpful in other diseases. And with the amount of resources it takes to develop gene therapy, if you can develop something that works for multiple different medical conditions, that can be really helpful in terms of funding and research and development. There was also a presentation for NF1 on a type of exon skipping using mice. And the reason that they did exon skipping, particularly exon 17, is we think that it's possible that part of the NF1 gene might not be completely necessary for the gene to function correctly, because there's never been a patient with NF1 that has a deletion of that part of the gene. But there are people that have small mutations within that part of the gene that do have NF1. And so the thought was, well, maybe we take out that whole piece of the gene, and that might kind of prevent the NF1 symptoms. And they've done it in mice, and it seemed to reverse the NF1 in those mice. And so they want to see if they can develop it further for people and affect all of those mutations within that part of the NF1 gene. And so they did some initial studies in mice, and they found that it worked in the mice a little bit, but not completely. And so they're going to keep developing it further. It got to some parts of the mice, particularly their brain, but not other parts of their bodies. This works on the RNA because it's exon skipping. So this is kind of like a temporary gene therapy that would have to be given continuously if it moved on to humans. But it is interesting in that it can be used for many different mutations in a very particular part of the gene, rather than just being custom to one particular person. Exon skipping might work for other types of NF1 mutations as well, particularly two other parts of the gene, exon 52 and 24, similar to 17, might not be totally essential for the gene to work correctly. So that this is a type of therapy that might be able to be used for different types of mutation along the gene. There was also a study on exon skipping in NF2, so the same technology, but with the NF2 gene. And the thing about NF2 is we do know that this type of mutation someone has can have a really big impact on how mild or how severe their NF2 symptoms are. And so truncating variants or kind of nonsense variants in the NF2 gene are more likely to cause a younger age of NF2 symptoms and more severe symptoms than someone who has a large deletion, even like a whole exon deleted. And so this study was investigating if they could take someone that has one of those more severe nonsense truncating mutations in exon 11 and then delete the whole exon and turn it into a deletion mutation, could it make a milder version of NF2? So not curative, because those deletions of a whole exon can still cause NF2 mutations in many people, but could it kind of modify the severity? So that's why they were investigating that. They were doing it on some cells, and they were able to see that it modified the gene, and that the gene with the deletion instead of that truncating mutation worked better. There was more RNA, there was more protein being made. But as I said, this is something that's wouldn't be curative for someone with NF2. So there's a lot of limitations for that option. There was one study presented on CRISPR style gene editing for NF1 on a cell culture. So very initial studies on cells where they went and they were able to correct a mutation in the NF1 gene and see that the NF1 RNA and protein improved, that more of it was being made. And we know that NF1 cells grow quickly and too much, leading to the tumors. And they were able to see that their cells stopped growing as much because they had functional NF1 again within them. So it was a really good, promising early study about that. It was a very specific type of CRISPR that only worked for certain mutations. So that's a limitation of that one. It only worked for a type of mutation where there's usually the base pair G and it gets switched to an A. The other thing about this type of CRISPR is though you have to target it very specifically to one section of the gene, within that section, sometimes you can add in extra mutations that are not supposed to be there. So a couple letters away, you could add in a mutation, and that could cause problems. So it's not a completely perfect way of editing a gene. And then lastly, there was a presentation about a type of suicide gene therapy for schwannomatosis. And what it does is it inserts a gene into the DNA that causes schwannomas to actually start to like shrink and the cells to die within a schwannoma. And it's something that's injected directly into the tumor. They did the study on mice that had schwannomas. And they looked at the mice and they could tell that the mice had less pain from their schwannomas after the treatment because there's different kind of behaviors that mice do when they're in pain. And they could see that the mice stopped behaving that way. And this is Dr. Brenner's research. He really focuses on the pain piece that impacts people with schwannomatosis. And because this is not specific to the specific mutation or even the specific gene, this is just kind of acting on the biology of schwannomas. It's something that could work for all types of schwannomatosis, NF2, LZTR1, SMARCB1. Even if we couldn't figure out which gene causes someone's schwannomatosis, it could potentially work. And it's something that's injected directly into a tumor, so each tumor to treat that tumor very specifically. So kind of a different approach rather than trying to treat the whole body. Instead, can we inject into each tumor and then cause the tumor to be less symptomatic? So these are kind of the highlights from the global meeting about where we're going with things. Very promising initial research on different gene therapy options, but we still have a long way to go with this. So we're still in cell culture stages, we're still in mice model stages, and it does take time for us to get to a place where we feel something's safe and it has the potential to be effective in order to start human clinical trials for these conditions. So that was the end of our slides, and we really love questions. So we would like to open it up to the floor. I'm going to stop sharing.
Wonderful. Thank you so much, Kara. Yeah, so I want to encourage anyone who has questions, feel free to unmute and ask your question, or drop it in the chat if you're more comfortable, and I'll ask it for you.
So one of my questions is this research only going on in the USA, or is it also in parts of Europe or something like this?
That's a good question. The stuff that was presented in June, the NF2 exon skipping research was actually a researcher from Spain was presenting on that. We do have a lot of our colleagues in Europe in particular that we see at all these meetings that are really global meetings for NF that are working on different things. I think it's really also dependent on kind of the research pipeline for gene therapy. So in the United States, there is actually a system set up for rare disease research and approval of rare disease therapies in particular, that is pretty robust. And so I imagine that a lot of the initial stages of gene therapy development is more likely to move along quickly in the United States because there's a lot of motivation for that. But I know we are a global world, and we know that NF and schwannomatosis impacts everyone worldwide. And so I know there's a lot of motivation to do site studies that would be global in nature so that everyone can access these things. But I think a lot of the resources we have are in the United States to move this along and get the funding for this kind of research.
Yeah, and just to add to that, I think any initial gene therapy trials in NF are going to be at least confined by country, just because the regulatory issues are going to be extreme. And so it would be a later move to try to have trials that were more globally based, because the red tape.
Oh, I think Miranda's frozen. That's
I think we
unfortunate.
froze her. I think if I can extrapolate from hers, because of the potential risks involved with gene therapy, and is it such a more novel treatment, it's possible that early studies are really going to operate within a single country, because to meet all of the what we call regulatory requirements and the ethics approvals in different countries can be really challenging because they have different rules around it. So it's something that probably I wouldn't be surprised if the very first human trials were within the United States, and then afterwards were in other countries.
So NF is so diverse. You know, if we just even look at NF1, NF2, or schwannomatosis, you can have people in the same family. Like, I know my experience with NF is wildly different from my mom's or my sister's. So how are they thinking of honing in on what area so that they're, how do they hone in on the area that's going to have the greatest impact on the greatest number of people, or even just the most success? What is the thinking there, and how are they getting there? That makes sense.
Part of that is driven by the science, right? What happens first? NF is challenging. It's a big gene. It makes a protein that is not even easy to dissolve to be able to do anything with it because it's so large. And so a lot of what will probably lead in the NF space is going to be based on what has early efficacy in the lab that is translatable in a reasonable way, right? So it makes a task for what we're trying to do, right? Inform patient communities so that everybody's ready for it, because we really have no idea what's going to come first. There's people working actively, and who knows what is going to be the first step that actually gets into a clinical space. So it's challenging in that regard, right?
Another thing is that we need to look at clinical features of NF and schwannomatosis that are measurable. So things that we can show that something is working in a clinical trial. And so a lot of clinical trials that we hear about in NF and schwannomatosis are focusing on a tumor becoming smaller, because that's something that we can measure on MRI, that we can measure with photographs. Other types of symptoms related to NF can be a little bit harder to get a kind of a discrete measurement for. So that can sometimes play a role in how studies are designed and what they choose to use as like the primary treatment outcome for a clinical trial. So usually there's a primary outcome. What's our number one goal? What are we using to say this worked? And then they might have what we call secondary, like, we'll see if this worked too, but this is not the primary reason we're doing the study. Something that might also play a role is looking at what is the impact of that medical problem on our patient community. So what has the greatest impact on causing disability or affecting quality of life or potentially shortening lifespan? What is a clinical feature that we don't have anything good to treat with right now? So what is the gap in treatment? So those sometimes do play a role. The science leads. If the science can't support it, we can't develop something. But these are some of the big questions that come into place is, where is the need and can we measure it right now?
And that's another thing that Kara and I have been working on as well, is the patient aspect of this, because when the clinical trials do get started, they need to enroll. There need to be patients that are interested in receiving these treatments, right? And so Kara and I are actually getting ready to open up a patient preference survey for gene therapy that Kate will be sending out through the registry. So if when you guys see it, jump on it, because that is what we want to share with gene therapy researchers, so that not only is the science leading and all of these other things like outcomes working out the way that they do, but that so we're focusing on things that meet the needs of patients directly and what patients are looking for.
Yeah, I appreciate you bringing that up, Miranda. So I want to, because we're getting close to the end of the hour, I want to take that as an opportunity to say just a couple of things. So first is that that's one of the reasons I was excited to have the Kara and Miranda speak today, and that CTF is fully on board with sharing the work they're doing via the registry coming up in a little while, is because of that commitment to involving the patient voice in these conversations. Gene therapy is a big subject. It can be intimidating. I appreciate everyone that showed up today and asked some questions and really tried to dig in. It's one that can feel like, well, is this question going to be stupid because maybe I should have understood something that I missed? And Kara and Miranda have done a wonderful job consistently showing up in patient spaces and trying to break down some of those barriers, which is so important. So having said that, I'm going to put my email in the chat, and I want everyone to know that if you leave this call today and you think, well, I did kind of have a question I didn't ask, or I thought of something later, I'm going to send this recording out so you might rewatch it and think of something. You're more than welcome to reach out to me anytime. I can pass that along to both of them, get you the answers for any questions that you're asking, because I know that they're with me and wanting to encourage these kinds of conversations and involvement from this community. So I'll put my email in the chat for that. And then other than that, I just want to thank Kara and Miranda again for being here today, for bringing this information to us and making this available to everyone. So thank you for joining us, everybody from RAINS and from CTF Engage. It was great to have you here. As I said, I'll send this recording out both to everyone who was here today, as well as those who registered, and then to the entire CTF Engage. And then I'll share it with Vanessa Merker, and she can make that available to the RAINS folks as well. So thank you, everyone.
Thanks for having us, Kate. And I just want to reemphasize, don't hesitate to ask questions. We want you to understand. So please send on the questions. There are no stupid questions, right? It's just things that help you understand more.
Absolutely. Thank you, Miranda. Have a good day, everyone.
Good one.